Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. You will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material.
In this video, we're going to go over some of the common organelles of a eukaryotic cell. Now, an organelle is simply a word that means small part. So just like the body has small parts called organs, cells have small parts called organelles. Well, there's a lot of them, so let's go ahead and get started. First thing I want to mention are the three basic sections to the cell. And all cells need to take in food, get rid of waste, and reproduce. Well, cells have three main sections to them, and inside of those three sections are parts that allow them to take in food, get rid of waste, and reproduce. So the first section you can see highlighted by the flashing black line, the plasma membrane. You might also hear the plasma membrane simply called the cell membrane. We'll talk more about this in a little bit. Pulsing in the picture, you can see the second main section, the nucleus. You often hear the nucleus called the control center. We'll explain why in a little bit. And lastly, we have the cytoplasm. Now, the arrow in the picture looks like it's kind of just pointing to empty space. Well, cells are not filled with empty space. They're filled with a part called the cytoplasm. We'll go into that in more detail. Let's actually do the cytoplasm first. Here we go. So the cytoplasm, the first thing I want to mention is that it's the jelly-like material on the inside of the cell. You know, when you look at diagrams, sometimes in a diagram a cell might look like it's empty or filled with hollow air space, and that's not true. Cells are filled with a jelly-like liquidy material called the cytoplasm, and in that cytoplasm most organelles float within. You can see here's a list of a few of the organelles that are floating around in the cytoplasm. We're going to go and talk about all of these throughout this video. We'll talk about what the mitochondria is, what lysosomes are. And because the cytoplasm is a jelly-like material and things are floating around inside of it, you often, under a microscope, can actually see parts floating around. In this animation here, you can see what's known as cytoplasmic streaming. The green circles that are drifting around inside of the square cells, the rectangular shaped cells, those green circles are plant cell chloroplasts. You can see in every rectangular shaped plant cell there's a few dozen chloroplasts in each of these cells here. And this is known as cytoplasmic streaming. Okay, well, what about the job? What does the cytoplasm actually do? Well, a lot of chemical reactions take place within the cell, and chemical reactions take place in the cytoplasm. So that's kind of what the job of the cytoplasm is. It helps to facilitate chemical reactions, and it also helps to dissolve solutes, particles that are in the cell, carbohydrates, proteins. These are all dissolved within the actual cytoplasm of the cell. So that's really what the main function of the cytoplasm is. It also adds support to the cell, a pressure support for the cell. So the next part I want to mention is called the plasma membrane, also known as the cell membrane. And first of all, its composition means what is it made from? It's a bilayer of lipids and proteins. Earlier in the school year, we learned about lipids. And this is a drawing of a very simple lipid here. The round part represents the head, the two tails are the fatty acids. And what we mean by a bilayer, it's not just one layer, but two layers on top of one another. This is a bilayer of lipids right here. In between the bilayer of lipids are also proteins. I've labeled this protein channel, and you're going to see its importance in a moment. Well, the job of the plasma membrane is to allow materials to to pass, to enter and exit. You know, small molecules like water and oxygen, H2O for water, O2 for oxygen, small molecules like this can simply pass through the lipid bilayer. However, larger molecules like glucose, have to pass through these protein channels, and that's what you see happening in the animation. And so that's really the job of the cell membrane. But the cell membrane is what we call semi-permeable or selectively permeable. And that means that it doesn't allow everything in and out. It only allows certain specific molecules and ions and charged particles to enter or exit through the plasma membrane. So when we come back to this picture we saw earlier, here we have the next part I want to mention. You can see the black square highlighting the nucleus. Let's zoom on in for a closer look. When we zoom on in, here we have the nucleus. And you've heard maybe in middle school that the job of the nucleus is to control the cell's activity. You often hear it given the nickname of the headquarters. Well, what does that mean? Well, the reason we call the nucleus the headquarters is because it contains DNA. And in the picture, we see 5 twisted strands of what is called chromatin. Chromatin are long strands of DNA. And DNA is the instructions, the blueprint, the manual on how to make proteins. And this is why we call the nucleus the control center, is because the chromatin will be sending out information to make proteins. And proteins do a lot of the work. The vast majority of the work inside of our cells, inside of our bodies, are performed by proteins. So this is why the nucleus gets the nickname of the control center. Also inside the nucleus there's a round object known as the nucleolus. And the job of the nucleolus is to make another part of a cell, another organelle, called the ribosome. So the job of the nucleolus is to make ribosomes, and we're going to see what ribosomes do in just a moment. When we look at this picture right here, we can see a nice electron microscope picture. The overall round object is the nucleus. You can see the dark spot is the nucleolus. A really nice picture here of the nucleus and the nucleolus. So we come back here, we have the box highlighting the rough ER and the smooth ER. Let's zoom on in and take a closer look at the endoplasmic reticulum. Okay, first of all, there's 2 kinds of ER. Let's look at the rough ER first. The ER stands for endoplasmic reticulum. The rough ER, the reason it's called rough is that it's covered in ribosomes. So we mentioned a moment ago that the nucleolus makes ribosomes. The ribosomes then exit the nucleus and travel along this tunnel system known as the rough ER. And that's kind of the job of the rough ER, is to receive the ribosomes and transport and carry around the ribosomes from one part of the cell to another. As you can see, we finally see what the job of a ribosome is. Protein-making ribosomes. So that's the job of a ribosome, is to make a protein. So the rough ER will transport the ribosomes as the ribosomes are making a protein. Well, if we look at this picture here, we have the purple nucleus. Identify part A in the nucleus. I hope you know that's the nucleolus. The lines, the purple lines identified by letter B, that's the DNA. I hope you remember the name of the DNA. That's called chromatin. And then the outside of the nucleus in picture C. I never mentioned the name earlier, but that's called the nuclear envelope. Notice how the blue is the rough ER, and there's gray dots all over it. Well, look in the animation. You see gray dots exiting the nucleolus in the animation. And those gray dots are traveling through the blue rough ER. That means those gray dots have to be the ribosomes. The job of ribosomes are to make proteins. We're going to go into more on that in just a few moments. So what about the smooth ER? Well, if the rough ER is covered in ribosomes, then a little common sense, the smooth ER is not covered in ribosomes. And you can see it has a different job. The job of the smooth ER is to help make lipids and fats and to help break down certain toxins. But the reason it gets its name smooth ER is it's not covered in ribosomes. The rough ER is covered in ribosomes, and that's why it kind of has a rough appearance to it. So let's talk a few extra moments about those things called ribosomes. They're created by the nucleolus. Remember, the nucleolus is inside the nucleus. They're transported. The ribosomes are transported by the rough ER, and as the ribosomes are being transported by the rough ER, we now come to their job, their duty. Their function is to make proteins. And so if we highlight a few ribosomes with that flashing black box, let's zoom on in to a ribosome that's attached to the rough ER. Well, here we have a ribosome, and the reason, what I mentioned a moment ago, is its job is to make a protein. And what it does is it will gather individual amino acids. And earlier in the school year, I hope you remember that amino acids are the building blocks of proteins. And so here we see a ribosome is gathering and connecting a bunch of amino acids into a long chain. This long chain of amino acids is what we call a protein, and that's the job of a ribosome, is to gather and build these amino acids into a larger protein. This process, by the way, is known as translation. We're going to learn about it later in the school year, though. So if we come back out here to our diagram, I've highlighted the flashing box of the Golgi body. Let's zoom on in to the Golgi body. You might often hear it called the Golgi apparatus or the Golgi complex. It's always going to be called Golgi because it's named after the scientist with the last name of Golgi who discovered it. And so the job of the Golgi body is to receive proteins that ribosomes made, modify them, sort them, and package them so they can be exported from the cell. And the Golgi body will put the proteins into a protective little envelope known as a vesicle. And so let's go ahead and review what we've learned really quick. When we look at the process of shipping out a protein, in our animation we can see the nucleolus is releasing a black dot. That black dot represents a ribosome. So the nucleolus makes a ribosome. Notice that ribosome then travels through that rough ER, that system of tubes and tunnels. So the ribosome is traveling through the rough ER making a protein. Well, let's zoom on in for a closer look. So there's the ribosome. Okay, now that we can see the ribosome, remember what the job of a ribosome is. It makes a protein. And so there you see the word protein. Now it's spelled P-R-O-T-E-I-N, but it's not really put together properly. So this protein is not quite finished, even though the ribosome made it. Well, watch what happens next. Remember, this is all happening at the rough ER. The protein is going to be placed inside of a protective vesicle and travel away. Where is it going? I hope you know where it's going. It's going to a Golgi body. And so the protein arrives at the Golgi body. What happens to the vesicle? The vesicle fuses and becomes a part of the Golgi body. Well, watch what happens. The protein moves through the Golgi body and is eventually processed and modified and ultimately is going to be released. So the Golgi body kind of makes the final changes to the protein. Well, when the Golgi body releases the protein, it also will put it in a protective vesicle. So the vesicle is going to be a protective little envelope. Watch this. The protein is put into a protective vesicle and it's released. The vesicle fuses with the cell membrane, and then the protein is simply released from the cell, goes where it's needed. All right, so if we were in class, I would have you turn to your neighbor and try to work on these review questions. So we've kind of covered a lot of information. Let's take a quick moment, pause the video, try to answer these 7 questions. I'm going to go over the answers in 3, 2, 1. So which organelle builds proteins? I hope you know that's a ribosome. Which organelle stores DNA? Well, that's the control center called the nucleus. Which organelle produces ribosomes? Well, that's the nucleolus. It's found inside the nucleus. Which organelle creates fats and breaks down toxins? Well, that's the smooth ER. Which organelle, number 5, packages and releases proteins from a cell? Well, that's the Golgi body. Remember, they're put inside of a protective vesicle. Number 6, which organelle allows materials to enter and exit a cell? Well, that's the cell membrane, also called the plasma membrane. Number 7, which organelle links chains of amino acids together? Well, that's the same thing as question number 1. Question number 1 and question number 7. It's the same question, just worded a little differently because amino acids make proteins. Let's try this one. Same thing in class. I would have you work on this with a neighbor for a minute, but pause the video. Try to put these 4 steps in order. What comes first, second, third, fourth? I'm going to show the answer in 3, 2, 1. Okay. So the answer is, I hope you chose step C first. The nucleolus makes ribosomes. And then I hope you chose D. Ribosomes will exit the nucleus. And then I hope you chose B. The ribosomes are going to travel along the rough ER and create proteins. And then finally, I hope you chose A. The Golgi body will package and export the finished protein inside of a protective vesicle. Let's move on. So now we're back to our diagram, and I've highlighted the flashing box, the mitochondria. Let's zoom on in for a closer look. Here we have the mitochondria. Artist drawing on top, actual picture on the bottom. And so the job of the mitochondria, you know, in middle school they give cute little nicknames to cell parts. In middle school you may have been told this is the powerhouse, but you may never have been told why. Well, it's the powerhouse because its job is to make an energy molecule by the name of adenosine triphosphate, ATP. So the mitochondria does a very important process known as cellular respiration. And during the process of cellular respiration, it will create an energy molecule by the name of ATP. ATP is a molecule used by your cells for energy. Okay, let's zoom on in. Let's focus on the lysosomes back in our diagram here. Let's zoom on in for a closer look at the lysosomes. And so the lysosomes contain inside of them very powerful digestive enzymes, and that's what my little animated scissors here are showing. So the blue circle is the lysosome, with the animated scissors representing the enzymes inside. And so lysosomes have various jobs. Number 1 is to break down food. So here we have a protein molecule that's being taken into the cell, and the protein is being broken down into smaller parts. I hope you know those smaller parts are called amino acids. Here we have an example of a white blood cell. You probably know white blood cells' job is to fight off infections like bacteria and viruses. Well, that's what we call pathogens. Things that cause disease are called pathogens. And so here's a virus or a bacteria, and notice how it just landed on a white blood cell. Well, what happens is the white blood cell will swallow up that pathogen, put it inside of a lysosome, and those powerful digestive enzymes will destroy it. So that's one way that our immune system keeps us healthy. And then there's the process called autolysis, where it's basically an aging cell will destroy itself. And so in the middle of our picture here we have a group of cells, and I have the animated scissors, because what happens is when cells are dying and aging, they will kind of self-destruct in a process called autolysis, where the lysosomes will release all the digestive enzymes and kill that cell. It leaves behind a gap, but don't worry, that gap will simply be filled in as cells multiply. So you've heard the expression, out with the old, in with the new. A couple other cell parts I want to mention are used in movement, and that's the job of cilia and flagella. Now, not all cells move, so not all cells have cilia and flagella. But if cells move, they probably have either cilia or flagella. So the picture on the left shows the cilia, short hair-like extensions. There's numerous, hundreds and thousands of them per cell. Way too many to count. Very short, stubby, almost look like eyelashes. They're so short and stubby. The picture on the right shows a sperm cell with a real long flagella. Flagella is actually the plural version. Flagellum is singular. And so the difference is really their length. The cilia, short, stubby. Flagella are long and whip-like, and usually there's only a few of them, maybe only one flagellum maybe 2, maybe 3, maybe 4, but not nearly as many as the cilia.
Organisms are made up of organ systems, such as the digestive system, pictured here. Each of these organ systems are comprised of individual organs. As shown in the diagram, organs such as the liver, the stomach, and the small and large intestines are part of the digestive system. All of these organs are made up of tissues. Tissues are a group of similar cells that perform the same functions, such as the epithelial tissue and the smooth muscle tissue pictured here. Tissues are made up of cells, and a cell is the smallest unit of life. Finally, cells contain organelles and molecules such as DNA. Let's examine a cell more closely. There are two general classifications of cells, prokaryotic cells and eukaryotic cells. Prokaryotes are single-celled organisms and include bacteria and some algae. On the other hand, eukaryotic cells make up more complex organisms like plants and animals. Eukaryotic cells contain a membrane-bound nucleus that holds all of the DNA in one part of the cell, as well as many other organelles. Now let's look at the function of each organelle in a eukaryotic cell. To gain an understanding of the inner workings of the cell, it's helpful to think of the cell as a mini city. Just like in a city, a cell is made up of many parts that have their own functions. In the cell, these parts are called organelles, which are explained next. A plasma cell membrane is made up of a phospholipid bilayer. The bilayer surrounds the cell, protecting it, and functions as a gate that allows molecules into and out of the cell. In our city, the cell membrane would be like a large fence surrounding the city that only allows certain molecules to go in and out of the cell. The nucleus is like the Capitol building of the city because it functions as the control center of the cell and houses the genetic information or instructions for what the cell is supposed to do. The mitochondria functions as the powerhouse of the cell by producing the energy the cell needs to perform its duties. This is just like a power plant providing energy for a city to use. The energy currency of the cell is ATP. The ribosomes act as factories which translate the genetic information, or DNA, from the nucleus into usable protein molecules. Proteins do the work in the cell. The Golgi functions as the post office in the cell and packages and modifies the proteins. The endoplasmic reticulum is the highway of the cell. It forms an interconnected network of tubules and vesicles and functions to transport proteins throughout the cell. So those protein-containing vesicles would be like the trucks on the highways of our city. Many of the protein-producing ribosomes are located on the endoplasmic reticulum. The lysosome is the trash man of the cell because it digests excess or worn-out organelles and proteins by breaking them down and removing them.
Let's talk a little bit about the structure of the cell. I've done a lot of videos where we deal with things that go on inside of them, but I haven't done one where we just talk about the entire structure of them. So a good place to start is, let me just draw the membrane. And the cellular membrane is a good place to start because this is what separates the cell from the outside world. And to a large degree, it kind of defines the cell. It defines it as this very, very small compartment. That's where the word cell came from. So let me label that. Cellular membrane. And all cells have a cellular membrane. Now, if we think about maybe the most important thing that defines a cell, you've probably seen in the DNA videos, and we're going to talk about translation and transcription and all of that, that what defines what a living organism is its DNA. So all cells have DNA inside of it. And I won't go into the details of how DNA defines what an organism is. I've done that in some detail on the DNA videos, but all cells have DNA here. This is more of a kind of an anatomy of a cell video than necessarily the function, but we'll go into the function because we need to know what these different parts do. So this is right here. This is the DNA. This is the DNA, and it's in its chromatin form here. There's also little proteins here. There's protein, not in all organisms, but we're going to stick to eukaryotes, and I'll talk a little bit about the difference between eukaryotes and prokaryotes in a second. But you have DNA. As I've drawn this cell right now, this is pretty much any cell in any animal or plant or whatever kingdom could look like this. I haven't drawn a lot of the details. I've just drawn the DNA and the cellular membrane. Now, here's kind of the first major division in the living world, or at least from our point of view, or that it seemed obvious, is that some cells have a membrane around the DNA. Around the DNA. So they'll have a membrane around the DNA that separates the DNA and the chromatin and everything that makes up the stuff within the DNA, separates that from the rest of the cell, and this is called a nucleus. This is called a nucleus. And I said that's a major division because when some people looked at some cells and they saw nucleus, and other cells and they didn't see nucleus, they say, hey, this is a good way to classify organisms. So they called things that had nucleuses eukaryotes. Eukaryotes. These have a nucleus. So as I've drawn this cell right here, it is a eukaryote. Now, if you do not have a nucleus, you are dealing with a prokaryote. Prokaryote. No nucleus. And examples of prokaryotes, the two big groups of them, are bacteria and archaea. And archaea are really interesting. We know very, very little about them. They were originally thought to be types of bacteria, but now people are realizing that they're this whole completely other group, and we've actually observed a very small subset of them. So it's a very fascinating group. And it actually turns out that evolutionarily speaking, you shouldn't make this division first, that it actually makes more sense to divide things into eukaryotes. I'll just write that, euc, bacteria, and archaea. That you don't want to do this division first, that there are actually three separate groups that you want to start off with. Archaea. But we'll do that. We'll talk more about this in future videos. But if you want to say who has a nucleus, well, eukaryotes have a nucleus by definition. Who does not have a nucleus? Well, the bacteria and the archaea do not have nuclei, just like that. But I'm going to focus on eukaryotes because they tend to be a little bit more complex. They tend to be larger, and most of what we talk about, at least in the videos so far, are dealing with eukaryotes. Eukaryotes include plants, animals. We're animals, at least I am. Animals and fungi, and there are other groups within eukaryotes, but these are the ones that we normally deal with in our everyday world. But let's go back to looking at the anatomy of cells. So we have our DNA. We know that it gets transcribed into mRNA. That mRNA leaves the nucleus, and it gets translated into proteins at the ribosomes. So the ribosomes are these little complexes that could be floating all over the cell. And we'll see in a second that they can also be attached to these other membrane structures. So this is a ribosome. Ribosome. And if all this talk of DNA transcription into mRNA and mRNA leaving the nucleus and traveling to the ribosome to be translated into proteins makes no sense to you, there are several videos where I go into that in detail. But what I want to do is just focus on all of the different parts to kind of give a big picture of things. So ribosomes are where mRNA that gets transcribed inside the nucleus from DNA, where it gets translated into proteins. So you can kind of view them as the place where information actually turns into proteins, which then can be used anywhere else in the cell. Now, one interesting, and these ribosomes, they're made up of proteins, and actually they're made up of RNA. And so one question is, well, where are the pieces of the ribosomes made? Well, some are made by proteins, which might be made in other ribosomes. But some of it, the mRNA ribosomes, you can kind of view them as this big, as this mess if you were to look detailed. There's some protein there, and I'm not drawing it that realistically, but then you have some mRNA tied in with the protein. And the mRNA isn't used for actual information purposes like it normally is when it goes from DNA to the ribosome. Within the ribosome itself, ribosomal RNA is actually used as part of the structure. It actually helps the ribosome function as a ribosome. So it's actually part of the ribosome. And all of that gets built in a part of the nucleus called the nucleolus or the nucleole. Let me write that down. So this right here, this is interesting right here. This is the nucleolus. the nucleolus or the nucleole, and it isn't a separate organelle, and it's not separated by a membrane, but it shows up in a microscope. So when people first saw that, hey, there's like a bundle there, that must be like the core of the nucleus or something. But it turns out what it is, it's a densely packed, you got DNA and RNA, and that's actually where the ribosomal RNA, the stuff that makes up the ribosomes, is actually produced. But it's so dense that it shows up on a microscope, and that's why people decided to name it something different. But it is not membrane bound. It's not an organelle within an organelle. It's just densely packed of proteins and ribosomal RNA. It's where ribosomal RNA gets produced. So anyway, we're at the ribosome. That's where proteins get produced. But if the ribosomes are just floating around, if they're free ribosomes, then those proteins will just, once they're produced at the ribosome, they'll just float around here in the fluid inside the cell that we call the cytosol. Cytosol. But what if we wanted to produce proteins that are supposed to end up maybe in the membrane of the cell, or maybe outside of the cell itself? Cells produce things that are used by other cells, that are used by the rest of the body. And here we have to go to proteins that are attached to this membrane. You can kind of view it as a bunch of tunnels. Let me see how well I can draw it. I'm going to draw it very roughly. You have this thing called the endoplasmic reticulum. Endoplasmic reticulum. You can just view it as a bunch of tunnels like that. Endoplasmic reticulum. And they eventually lead to something called Golgi bodies, named after Mr. Golgi himself. So I'll do the endoplasmic reticulum in yellow, and I'll do the Golgi bodies. I'll do the Golgi bodies in green, maybe, just like that. And I'll tell you in a second what they are. So this is kind of a big kind of stack, or you can kind of view it as a bunch of folded membranes together. And some ribosomes are actually attached to this part, which I call the endoplasmic reticulum. So we have ribosomes attached. Some of them are free, some of them are attached. And let me write my labels down. So this right here, and we use the space over here. This big flap of convoluted membrane, that's the endoplasmic reticulum. Endoplasmic reticulum. It's fun to say. Endoplasmic reticulum. Maybe a good name for a band. Endoplasmic reticulum. And the parts that have ribosomes attached to it are called the rough endoplasmic reticulum. Maybe even a better name for a band. So right here, where I've attached ribosomes, so these ribosomes are attached right here. This is the rough endoplasmic reticulum, or maybe the rough ER. The rough ER, ER standing for endoplasmic reticulum. And then where we don't have any ribosomes attached, that's the smooth endoplasmic reticulum. So right here, that is the smooth endoplasmic reticulum. And I'm going to tell you in a second what this is, but we can keep following the membranes. Eventually, we get the Golgi bodies. Golgi bodies. And now what happens is, I gave you a hint. In our free ribosomes, mRNA gets there, gets translated to proteins, and the proteins then just float around in the cytosol. But what if we want proteins that should end up in membranes, or that end up outside of the cell? And that's where the endoplasmic reticulum and the Golgi bodies come into play. Because now what we can do is, we have mRNA coming outside of the nucleus, and it can attach to the ribosomes, or it can be translated by the ribosomes in the rough ER. And what happens here is your mRNA comes here. It's getting, I know I drew that arrow very small. It's getting translated on the outside of the endoplasmic reticulum. But as the protein gets produced, it's getting pushed into the inside of the endoplasmic reticulum. And when I say the inside of the endoplasmic reticulum, I'm talking about this area. I'm coloring it in. This is the inside of the endoplasmic reticulum. And so the proteins will get pushed into the endoplasmic reticulum, the ones that are to be used outside of the cytosol, outside of the cell, or maybe in the cell's membrane. So proteins will end up here. That's why those ribosomes are on the membrane, because they can translate things that are outside of the endoplasmic reticulum. But as the proteins get produced, the chain of the amino acid chain ends up inside of it. Let me make a blow up of that, because I think that'll be useful. So let me draw an endoplasmic. Let's say this is the membrane of the endoplasmic reticulum. And then you have ribosomes attached to it. So let's say that's a ribosome on the endoplasmic reticulum. And this is going to be the rough endoplasmic reticulum. And what you can have is mRNA coming into one side of it. Get your mRNA. mRNA can kind of come in through here. Maybe it's going in that direction. It's getting translated into the proteins. But then the protein, as the amino acid chain is built up, will pop out on this end of the membrane. Remember, this is our membrane of our endoplasmic reticulum. So even though the mRNA is on the outside, because the ribosome is attached to it, the protein can show up on the inside. And so once the protein is built, maybe it folds all up. Protein is just a folded chain of amino acids. It can travel through the endoplasmic reticulum. And it travels through it. It travels through the smooth endoplasmic reticulum all the way, and it gets to the Golgi bodies. And all sorts of other things happen. I'm oversimplifying things, but I just want to give you a sense of what everything in the cell does. And once the protein travels to the Golgi bodies and they get ready for traveling outside of the cell, or maybe traveling to the cell's membrane, they'll actually bud out of the Golgi bodies. So let's say the same protein, when it gets to the Golgi bodies, remember, it's inside of the Golgi body. So let me draw the membrane of the Golgi body right there. The protein might end up there. It's just a big chain of amino acids. And then it will bud out. So let's say it looks like that. And then maybe the next step, it'll look something like this. It'll look like that. It'll look like that. And in the very next step, you can imagine it'll look something like this, where it's actually completely budded out. It's popped out a little of the membrane of the Golgi body with it. So now the protein is surrounded by its own little membrane. Right? So let's think about what happened. We had DNA transcribed into mRNA. mRNA goes to a ribosome that's attached to the endoplasmic reticulum. It is translated into a protein that travels through the endoplasmic reticulum. First the rough, where all the ribosomes are, then the smooth. The smooth has other functions. It also helps, I guess we could say, produce hormones and other fatty compounds, but I won't go into detail there. But it just travels, it connects to the Golgi bodies. And the Golgi bodies, the proteins can bud off and take a little bit of the membrane with it. And this idea of something being surrounded by a membrane and just traveling in the cell. So maybe the protein now looks like this. I'm enlarging it. Maybe the protein's there, and then it took a little bit of the Golgi body membrane with it. This is called a vesicle. And this right here, let's add another one right here. I'm just doing that to label it. That's called a vesicle. And a vesicle is really just a very general term for anything, little small things, mostly proteins, that in the cell that are just floating around, that are surrounded by their own little mini membranes. And the reason why this mini membrane is useful is now this protein can now float to the outer membrane of the cell. It can also float to other parts of the cell. I am doing a simplification. And then it can merge either with the membrane of the cell, or it can use this membrane, this little vesicle membrane itself it has, to facilitate it getting out of the cell. You can imagine that it eventually, this thing could, let's say this is the outer membrane of the cell, and I'm doing a gross oversimplification. I'm not even drawing the bilipid layer, but just to have a visual impression of what it might look like. That's the vesicle there, the little protein inside of it, and it gets closer and closer to the membrane. And then it can merge with the membrane, because it's made up of the same stuff. It merges with the membrane, your protein inside. I switched colors arbitrarily. But now, all of a sudden, once it merges with the membrane, then the protein can exit the actual cell, or maybe it can actually embed itself within this membrane, within the outer cellular membrane, which I drew very thin, but it has 2 layers to it, and we'll talk more about that I could probably make a whole video on it. So those are, we've already made pretty good progress on showing the anatomy of the cell. There are a few other things we can throw in there. There are things called lysosomes, which are really, there's things called lysosomes, which exist in animal cells that contain enzymes in them that help dissolve other things. Lysosome. So if a lysosome attaches to something else and is able to spew its enzymes into it, it usually kills it. It usually digests it. So that's what a lysosome does. In in plants, you have things called lytic vacuoles, and those are really the same thing as a lysosome in terms of their function, in that they're really big vesicles. And in fact, in general, a vacuole is just a big vesicle. It's just a general term for a big membrane-bound organelle. Vacuole. And once again, what's an organelle? Let me write down that word. Organelle. It's just a membrane-bound, I guess you could call it, subunit of a cell. Just like my liver is a subunit of cell, an organ, and it's an organ. An organelle is a subunit of a cell. So a vacuole is just a general term for a membrane-bound organelle that stores stuff inside of our cell. So this stores things. So a lytic vacuole would be a vacuole in a plant cell that stores a bunch of enzymes, and if it were attached to something else, it would dissolve it if it were able to spew its enzymes into the other thing. Now, there's several organelles that we've talked about within the context of respiration and photosynthesis, and I go into detail in those videos. But we have things called mitochondria. Mitochondria in cells. And they have inner and outer membranes, and this is where we produce our energy, where sugars are turned into ATP. And I've done detailed videos on this. And these are actually contain their own DNA, and they can actually reproduce on their own, which makes a lot of, so they have their own DNA, which makes people believe that they exist, that their ancestors once existed as independent prokaryotic organisms, that once, at some point, they just kind of stumbled upon the notion of, hey, why don't I live it inside other organisms and kind of live in symbiosis? So mitochondria, these are organelles that at one time their ancestors might have been independent prokaryotes. So those are mitochondria. Let me write it down. Mitochondria, that's where cellular respiration takes part, and we go into detail on that. And then on the plant cells, this is only in, well, definitely not in animal cells, you have chloroplasts, which are a subset of thing called plastids. But chloroplasts are the most famous. Maybe I should do that in especially green. So we have chloroplasts, and we know they have little thylakoids in there. This is where photosynthesis takes place. You have your grana and all of that. And I do go into detail in the photosynthesis videos, but it's good to know these are other organelles. And just like mitochondria, they have their own DNA and their own ribosomes. And so the belief is that they were once independent prokaryotes that learned to live in symbiosis inside of larger eukaryotic cells. So we're almost done, really, with the structure cells. There's other things we can throw on here. If we're dealing with plant cells, or really non-animal cells, we'll have something called a cell wall, which will give some strength to the outer membrane. You can kind of view it that way, or you could kind of give some rigidity. So you have things called cell walls. So they're not necessarily completely rigid. You can almost view them as balloons that provide just a little bit more rigidity. Things like wood actually have double cell walls for really hardcore rigidity. So this is a cell wall. Cell wall. This is in non-animals. And in plants, it's made out of cellulose, not cellulite. Cellulose, that used to confuse me. So this gives extra rigidity or form to the cellular membrane. And then to actually give the cell its actual structure, you have these things called microfilaments, or sometimes actin filaments. And these are these little pipes that go throughout the cell. And these actually help give the cell its actual structure in 3 dimensions. And actually, they actually can participate in things moving around within the cell, or even the cell moving around itself. And just to be complete, make sure we're covering everything. If you watch the mitosis and meiosis videos, you have these things called centrioles. And I go into detail there. Centrioles that are right outside of the nucleus. 2 centrioles that are at right angles to each other make up a centrosome. And they kind of coordinate the microtubules when we start splitting the cells in mitosis and meiosis. So I won't go into detail there. I've done many videos on that as well. But so far, this is pretty much everything you need to know about, or at least a first overview of the structure of the cell.